we're going to be doing life sciences this afternoon. We're here for 3 hours. So hopefully within this period, I'm going to prepare you as much as I possibly can so that you walk into that exam room tomorrow and you just slay the dragon, because that's what it's about. It's walking in there with confidence. Knowing your work is 50% of it. The other 50% is actually understanding what the question asks you and answering what is asked of you. So you and I, this afternoon, we're going to be working together and we are going to do really amazing stuff. As we start though, see I'm old, I need my glasses, but as we start, what I did was I made a list of the sections that you need for this, for paper 1. And just little tips and things that I need for you to go through and make sure that you're covering. For example, the structure of the DNA and the RNA, you must be able to tabulate the differences. You need to know the difference in the function between DNA and RNA. And remember, there are 3 types of RNA. There's transfer RNA, messenger RNA, and then ribosomal RNA. You must know DNA replication. Protein synthesis, what they probably do is give you a question with a diagram, and you need to know that your transfer RNA, your messenger RNA is going to have the codon which goes from the DNA and reads the DNA codon and then moves out to the ribosome. All right, what happens? You need to understand the difference between transcription and translation. It gets asked in every single exam that's been written so far. Translation and transcription. All right, genetic engineering, you must know the risks and the benefits of genetic engineering. With regards to genes, chromatids, chromosomes, you must know what the diagram of the chromosome looks like. That you have your 2 chromatids joined by a centromere, and that you then have genes. You must know what a nucleotide is and what makes up a nucleotide. You also need to know meiosis, the different phases. The phases that are very popular are anaphase 2 and metaphase 2. So make sure that you're au fait with that. Just remember that when looking at meiosis, you've got 2 processes. The first phase is reduction division. All right, you're reducing from a diploid to haploid gametes. The second phase, or meiosis 2, is where you are duplicating. So you always end up with 4 unidentical daughter cells. Okay, that is the result of meiosis. Very important is during prophase 1, you are going to have the crossing over. So you'll have the chiasma, and that is the crossing over which results in genetic variation. So that is what makes us look different from our mom and our dad. So we don't just look like our mother and just look like our father, or you'd have an entire family that all look the same. That is why, because of the crossing over that occurs during prophase 1. Okay, then what a karyotype is. Okay, if a given human, in a given human genome, you must be able to identify the male and the female. Now remember, male will have an XY, 23rd chromosome, and your female will have an XX. And then if it's a Down syndrome child, chromosome 21 will have 3 chromosomes. Production of sex cells and the gametes, you must know that. So you must know what gametogenesis is, and be able to identify spermatogenesis from oogenesis. And then ethics with regard to cloning and genetically modified foods or organisms, you must be able to pass comment on it. If you are asked to provide your opinion, just make sure that you back it up with facts, all right? Then inheritance and genetic disorders, you must know your definitions. For example, you must know what variation is, you must know what alleles, what heterozygous versus homozygous is, you must know what recessive versus dominant genes are, the phenotype, the genotype, you must know those terms. Learn the law of dominance and the law of dominance and segregation. You need to know what a monohybrid cross is and how to do that monohybrid cross. I suggest you use, if you have to show any kind of cross, use a Punnett square, because you can't get confused about the lines that are crossing each other. Also make sure that if you are given a pedigree chart, that's a diagram where you've got your different generations. Each line is one generation, and what you have is the different characteristics that would come through. So let's say you'd have the parents, and the parents produce these children, which produce those children. So they've got bent fingers or straight fingers, or they can curl their tongue, or they have looped earlobes versus straight earlobes. All of that. Have a look at what is recessive, and then mark all the recessive organisms, those that carry the recessive traits, because they're easy to identify. And then, of course, in order to produce an offspring that has two recessive genes, so they're homozygous recessive, remember that one gamete has to come from each parent, which means the parents are then either heterozygous, or they must be homozygous recessive and homozygous dominant to produce if they're homozygous dominant, they can't. Sorry, it must be heterozygous or homozygous recessive to produce an offspring that has two homozygous recessive genes. So work backwards. Then with human and plant reproduction, please know how to label the male and female reproductive system. Also the sperm cell, make sure that you can label it. Remember, the acrosome is at the front or the tip of the sperm cell, and it produces enzymes which then penetrates the cell membrane of the little egg cell, so that the sperm can actually enter the egg cell and fertilization can take place. Make sure that you know the ovarian cycle and the menstruation cycle. Your menstrual cycle, your first 5 days of the cycle will be when the lining breaks away, the endometrium lining. And that is when menstruation takes place. And then, of course, you're going to have an increase in estrogen, which is to build the lining of the endometrium in case fertilization does take place. Because if it does take place, clearly you need a nice thick glandular, well-supplied, blood-supplied lining so that the egg can implant and, or the fertilized egg can implant and then grow into a fetus. Alrighty. Developing the fetus inside the uterus, there's a diagram which shows the development from the time it moves via the fimbriae into the fallopian tube, and then it shows the different stages where from fertilization as mitosis now occurs, because remember, mitosis is for growth and repair and replacement, and meiosis is for sexual reproduction. So we have the two haploid gametes. They have fused to form a diploid zygote, and then clearly the cell now has to undergo mitosis so that it can grow. All right. Then also prenatal and postnatal care, make sure you understand that. Human fertility, what increases or decreases fertility, just make sure that you've read through that. What they may do is give you a sort of a paragraph type question and then ask questions on the little paragraph. Sexual and, hang on, please make sure that you can discuss either being pro-abortion or anti-abortion. It comes up regularly. And then also all the various contraceptive methods. You must know your natural methods, your mechanical methods, your chemical methods, you know, so that you can explain that in an essay type question. The benefits of the different types of contraception, how effective they are, etc., etc. And also remember that any kind of barrier type contraception will also work in preventing the spread of sexually transmitted diseases. People then also, sexual reproduction in plants. Don't forget sexual reproduction in plants. With plants, remember, you've got your asexual reproduction, which is where a plant will grow a vegetative structure, which can be cut and then planted. So either the root or the stem, etc. But If you have sexual reproduction, we need male and female gametes. Now, the male gametes are produced in the anthers, the female gametes are produced, the egg cell is produced in the ovary, and then the pollen will then land on the stigma and grow down. But I'm sure there are learners with questions on plant reproduction. Make sure that you know the gynoecium of the female part or the gynoecium of the flower and how the little pollen grain grows down. And we then have the two male gametes because there's double fertilization. And please be able to discuss the double fertilization. Then also the diseases, all the different types of sexual diseases that your STDs that humans can get, know what they are and know how to prevent them. And then please, in vitro fertilization. Out of the exam papers, there are 8 available exam papers for the syllabus as it is now, for the grade 12 syllabus. And every single one of them has something on in vitro fertilization. Please make sure that you know what in vitro fertilization is. And I think that's just about it. That's paper 1. So this is what we're going to go through. Please also make sure that you take a calculator with you. So many learners seem to think we're writing life science. So it's not maths, it's not physical science, it's not accounting. I don't need a calculator. Yes, you do. Please make sure that you have a calculator that you can take into the exam with you. All right, you must be able to see the difference or determine the difference of. So they'll give you a whole bunch of readings for the one thing and the other thing, and you then have to tell the difference between the two or calculate the percentage of the difference. So you add, you subtract the two totals, and then you calculate the percentage, which is the total, that amount divided by the total times 100/1. You must be able to do those calculations, people. With regard to graphs, please be sure that you are able to draw a line graph and that you are able to do a bar graph, that you are able to distinguish between a bar graph and a histogram, and that you know what a pie chart is. And when we come back after the break, I'm going to go through a question which was in one of the exam papers. I can't remember. I just asked John to put it on the screen so I can show you how to do a graphing question. If you are able to graph and you are able to do basic calculations, I promise you now you have 25 to 30% in your exam. So don't throw those marks away. Make sure you take a calculator in, and I'm going to show you what you need to put on a graph in order to get marks. All right. So with regards to also your pie chart, whenever you do a pie chart, make sure that you either indicate on the pie chart with different shades and colors and little stars or circles, but so that you indicate the different divisions, or else you need to do that and then do a table or a key at the bottom, which indicates what each process or each of those colors that you used are. The same if you do a bar graph and you're comparing 2 or 3 different types of things. Make sure that you use different colors, and you must put a key, because the key then tells the examiner this is what each one indicates. Otherwise, it's all the same and it actually means nothing. If you have a line graph, and that line graph, you have 2 processes or 2 factors that you are graphing, use a dotted line and a straight line, or a red line and a blue line. Use a key to indicate what the difference is. For example, when you read your questions, have your pen in your hand and underline what is being asked, because very often learners answer the right answer to the wrong question. So please make sure that you actually read your question, read it again if you have to, and then answer it systematically. All right, but we're going to go through all of these exam techniques while we are going through the questions that our learners are sending through via the internet and also phoning through. Go to info@learnextra.co.za, and all the exam papers are there for you to work through. Okay, we're going to go across to the studio for an ad break, and I will see you in approximately 15 seconds to a minute. All right, see you just now. Going to just go to this bar graph question. It was taken from the February-March 2008, so it's the February-March 2009 Sept paper. And it sort of shows everything that I want to show you with regards to dealing with a graphing question. All right, if we have a look here, it says, records of human fertility over the period of 1941 to 1990 have shown changes in the sperm counts of normal men. Okay, now remember they're telling you it's normal men. The table below summarizes the changing percentages of men with a high sperm count and a low sperm count. All right, now remember this is in percentages and it's telling you from 1941 to 50 we have that 50%, and so men with high sperm counts start to decrease and decrease until 1981 to 1990, we're sitting only with 15% men with a high sperm count. But look at this, your men with low sperm counts increase. So there is a sort of an evening out here, but look at your table, look at what they're giving you and the information they're giving you. This is relevant. All right, now, the first question, by how much did the percentage of men with a high sperm count decrease in the period 1951 to 1970? So we're looking at this here. All right, first thing you need to do is you need to say 45 minus 23 is, come on, 17. But 17 what? It's 17%. Don't forget to write the unit down, people. Everything, any answer that you have to give a value to, please write the amount and the unit. So 10 kilograms, 15 millimeters per second. Always write the unit because sometimes they give you a mark for the number and the unit. Just in case they do, make sure you put both down. Okay, on the same system of axes, now this is the question that counts 11 marks, and it's easy, that's a definite 11 marks. Draw two sets of bar graphs, and they tell you, bar graphs, underline it so you don't think it's a line graph, to compare the percentage of men with high sperm counts and low sperm count from 1941. So let's extend our page here. first thing that you are going to do is you're going to give a heading. All right? That's number one. So they give you the heading in the question. They say to you, it is a comparison, because they say compare. So it's a comparison between percentage men. Oh, this pen's very difficult to write with, so you're just going to have to bear with me. I sometimes try to write too fast. Men, all right, with high sperm count and low sperm count from 1941 to 1990. That is exactly, if we go up, it's what the question asked. Compare the percentage of men with a high sperm count and a low sperm count from there. So all you're saying is, instead of compare, you're saying comparison between. And it's as simple as that. All right, now, let's look at our graph that we are the graphing. First thing you're going to do is you're going to put it down. All right, we're going to go here, we're going to go 10, 20, 30, 40, 50, 60. We don't have to go more than 60, because if you look back at our question here, it only goes to 50 anyway. So 60 is fair. All right? Now, something else, our years. Our years are, we've got 1, 2, 3, 4, 5 groups of years. So we're going to put them down here. We're going to say, right, we're going to go this to there. It's 1, 2, 3, 4, and 5. Okay, this one here was, okay, I've got the question next to me, so 1941 to 1950. We've got 1951 to 60. We've got 1961 to 70. And we've got 1971 to 80. And we have 81 to 1990. Those are our different processes. Okay, now, going back to our question, it tells us here. Now, remember, we're comparing high sperm counts and low sperm counts. So we do the high sperm counts first. So going down here, we're going to do, using a ruler, we know that it goes to 50, so it's to there. So this is our first, this is high sperm count. Okay, the next one is 42, I think. Let's just go back. It was 41, 40, 45. All right, so we go approximately 45 is going to be about there. Please use rulers to do all of this. I'm just estimating here at the moment. The next one was 28, so it'll be approximately there. 1971 was, I think, 21. 21. So 21 will be just over the 20 mark. And then our last one is going to be 15. All right, now you can either use a different color. I'm going to use a nice bright pink. People, let's just go to a break. I've got a bit of a technical problem here. And once we sort it out, we'll come back to you and we'll finish this question. Okay, this is typical Johannesburg. We had a power failure, shortage, whatever. Everything I just did the last 15 minutes is gone. So what I'm going to do is I'm not going to waste time going through it all again. What I'm going to do is show you that you needed to draw a bar graph, what needed to be represented, and where you get your marks from. All right, so let's go back to our question. I'm not going to do it all in detail, but I'm going to show you. Okay, this question here, on the same axes, draw two sets of bar graphs. So they're telling you on the same axes, always, people, bottom here will always be time. Okay, on this side, in other words, the vertical side, which is the your dependent variables are going to be along here. Here you are going to put sperm count in percentage. Okay, sperm count in percentage. Make sure that across the top here you go in numbers of 10, which is easy there because you go to 50, maybe go to 60, so that it's not at the top. The time along here, you're going to have your 5 periods, which are the 5 periods here. When you draw your different bars, I'm not going to do them, you're just going to put your bars in like that, etc. You're going to then choose a different color and you put your other bar in along like that. Please put in a key and don't forget your heading. Because if you put the heading in, the heading is immediately, first of all, the fact that you've got the correct type of graph, 1 mark. The fact that you have a title to the graph, 1 mark. The fact that you've labeled your x-axis correctly, another mark. The fact that you've labeled your y-axis correctly, another mark. So that's already 5 marks before you've even done anything. So there is no reason for anyone to get anything other than a full marks of 11 for any kind of graph question. Then when it says describe the trend for men with low sperm counts and compare it with those with high sperm counts over the 50-year period, so it's this entire period here, what can you see here? Look at what the information tells you, and it says men with high sperm counts, the sperm count has decreased systematically over 50 years, and men with low sperm counts, the percentages increase systematically over a 50-year period. And that's it. It's what do you see? Gives you another 2 marks. There's no reason for anyone not to have got 14 marks for this question, which is full marks. All right, I have a caller on the line. We've got her question on the board here so you can all see it and we can work through it. It's on protein synthesis. Hello? Not yet. Okay, we're just waiting for the young lady, not girl, the young lady to come on board. But just having a look at this question. It says, study the diagram below which shows protein synthesis. Now people, take your pen and paper and underline protein synthesis. What this does is it says to your brain, oi, pull that drawer out, take the file on protein synthesis out because that's what we're going to need in order to answer this question. That's number one. Number two, look at the labels and label everything that you can on the diagram before you even look at the questions. So if we look at A would be the nucleus. If you really want to be technical, you could say the nucleoplasm. Okay, it's inside the nucleus. All right. My learner's on the line. Hello?
Hello.
Who am I speaking to?
My name is Janine.
Is?
Janine.
Honey, I can barely hear what you're saying.
My name is Janine.
Janine, where are you calling from?
From Gauteng.
You from sunny, ha ha ha, but sunny Gauteng. It's so miserable today, it's not even funny. I feel like I'm living in the middle of London. But
yeah, very miserable.
Okay. Are you clearly battling a little bit with protein synthesis?
Yes.
Okay.
Very much so.
All you have to remember is every single person on, and I've got an echo here that's very off-putting, maybe the guys can fix it, but every person on this earth has a very, very special set of proteins in their body. Okay, that's why if you had a kidney problem and I wanted to give you a kidney, the chances that my kidney will actually be accepted by your body and not rejected are very, very small. You would have to go not even to your parents, but to your siblings, so brothers and sisters, for a closer match. Because remember, the closest your family can ever be is 50%. All right, does that make sense?
Yes.
Okay. Now, your body, with its very special sequence of proteins, it has to make those proteins which are perfect for your body and specific for your body. All right. Now, bearing that in mind, the recipe, and I mean, I know I'm oversimplifying this, but the recipe for proteins for your body, those recipes sit in your DNA. Okay? Now, before you do any question on protein synthesis or DNA replication, I want you to, on the side, write this. You write A equals T, and G equals C. And then you put a little dash here, backslash, and you put a U. And you make sure that wherever you are answering a question on protein synthesis or DNA, you are going to write that so that you never, ever forget that adenine always, its complementary base, it will always bond with thymine, and guanine will always bond with cytosine, only, ever. Okay, you will never get any other combinations. And if it is RNA, then it will be uracil. All right. So all your T's, if it's RNA, all your T's will be replaced with the uracil, or the U for uracil. Does that make sense?
Yes.
You happy?
Yes.
Okay, so that's number one. So looking at this, first thing we're going to do is you're going to put labels on. So we've got A is the nucleus. Now if you look at B, B is the actual ribosome. And the ribosome is where the protein is produced. If you remember from grade 10, when you did all the different organelles in a cell, there you learned ribosomes. And what is a ribosome's function? Protein synthesis. Okay, so it's its job. That's where it's going to happen. All right, this little hole here, there's your nuclear membrane, and that's the nuclear pore. And that allows for movement of your RNA backwards and forwards through from the cell cytoplasm to the nucleoplasm. Okay, happy? Are you happy, hon?
Yes.
It's with me so far. Okay.
Yes.
You must stop me. If there's something you want to ask, just ask me, all right?
It's
Okay.
mainly like transcription, how to explain
No,
it.
no, no,
in
no, no, no,
translation.
no, no. We haven't even got there yet. Let's
Okay.
first label. Anytime you get a diagram, you label it.
Yes.
I'll go through this whole process in detail, and then we'll look at the questions, okay?
Okay, thank you.
All right. So you've got the ribosome, and you've got the nucleus. Here's your DNA. All right. Here you're going to have your ribosome, which is the site for where this protein synthesis is going to happen. Okay, if we look at C, C is going to be your messenger RNA. And your messenger RNA goes along here and it reads what's on the DNA, and it then goes off to tell the ribosome, here's my combinations coming from the recipes from the DNA, and we now need to make this recipe. And the ribosome does that, if that makes sense.
Yes.
Okay, so that's our messenger RNA is C. And you'll see everything here is in threes. So this little thing here is called a code on. It codes on, or we can call it a triplet base, or a base triplet. It's nitrogenous bases, and it forms a triplet. They're three. Okay, so either a base triplet or a code on. I like code on because it codes onto. All right, now, that was B and C. Now, if we look at D, D is going to be a whole set. It's a little set or a recipe that's been produced. And D would, in that case, here, is going to be the recipe for your amino acid. So, and it sits here on E, which is your transfer RNA. Okay, so this here is your anticodon. In other words, it is the opposite to your codon. If you look here, this codon is GGG, and this one is CCC. It's the opposite. So that's the codon. This is the anticodon. The anticodon sits on the transfer RNA, and your codon sits on your messenger RNA. Okay, Janine?
Yes.
All right. And then if we look at this here, F is going to be the amino acids that are formed. And now if I take a whole bunch of little amino acids and I join them together, they end up making a protein. All right. Happy?
Yes.
Okay, now, what happens? The whole process is the recipe sits on your DNA. And I promise you, okay, you can't write that in an exam. They're going to laugh at you. But if you have that in your head and you understand that this code for your proteins sits on your DNA. So first thing that's got to happen is the DNA has to open. All right? So the weak hydrogen bonds that sit over here, you see, the little weak hydrogen bonds that are holding your nucleotides. Remember, a nucleotide is a phosphate, a pentose sugar, and a nitrogenous base. All right.
Yes.
That's one nucleotide.
Yeah.
Okay, so here's one nucleotide sitting there because they haven't drawn the phosphates and the pentose sugars as the spine or the backbone. They've only drawn the nitrogenous bases that you can see. Now, those weak hydrogen bonds break, and as they break and separate, so your messenger RNA can come on and it can copy and duplicate what is on this strand over here. You see? Now, the messenger RNA, because what they've drawn here, I mean, this is your messenger RNA. And this is one strand, so it's 1/2 of the DNA. Does that make sense?
Yeah.
So your DNA is opened, your messenger RNA comes in, and it forms complementary bases with what the information that you have on the DNA. Now it slips out of here, and it's all in threes, so each one of these is a codon, okay? And it slips out of here, and it goes to the ribosome. So here at the ribosome, and I hope they're showing you what I'm indicating here, but here you've got the ribosome, there's your codon. So where the DNA said CCC, we've now got GGG. Where it said on the DNA TAA, what have we got? The complementary base for thymine is going to be adenine. And then for adenine and adenine is going to be uracil. Because remember, with RNA, there is uracil and no thymine. Are you okay with that?
You said, isn't that a DNA inside the nucleus, not an RNA?
You have DNA, yeah. But, okay, what they've done is they've shown you step 2 here.
Oh, okay.
Okay, step 1 would be you'd have your DNA here, and you'd have your nitrogenous bases. Okay. All right, so whatever they are. So there's your DNA. So what happens then, this is step 1. Your DNA opens up, so it starts to do this. You understand?
Yeah.
There and there. Your second step is where your RNA will now come, your messenger RNA comes in here, and it will now, let me just extend this page, and we'll get another messenger RNA coming in here. And this messenger RNA is now going to form that, which is what you are seeing in our diagram. You follow? You're not seeing this part of the DNA. There's your double strand of DNA. And it opens up, and then your messenger RNA comes in, and so in other words, your other strand of DNA is going to be here somewhere. Let me do it in a different color. Your other strand of DNA is going to be here, but it's completely opened up now to make space for the messenger RNA to come in. Do you understand that?
Yeah.
Okay, so you're looking at step 2, not step 1 where it was one single strand.
Oh, okay.
Or at least one double strand. You happy?
Yes.
Okay. Now, Janine, it duplicates, and you've got your codons that are sitting on your messenger RNA. So it is the opposite. And this process of coding is called transcription. Now, if you look at the word transcription, trans means moving. Scription or script means to write. So it writes the opposite and then moves away. Another way of thinking of it is, do you remember, look, I mean, you're young. You're what, 17, 18 years old. But when I was a young person, which is a hell of a long time ago, we used to have cameras that used to take a photo on a spool, all right?
Yeah.
And then you'd take this little spool out and you would take it into developers, and they would then develop those photos. But first they'd make negatives. And then, okay, you'd have your negatives, And then your negatives you'd use, and then the guys in the photo lab would then use the negatives and make a positive, which was the photo that you had taken. That would be the thing I printed on white with colors, etc. All right, so now you've got that. This is like doing a negative. So it's doing the negative of the photo of the DNA. And then when the messenger RNA goes to the ribosome, your transfer RNA does the photo of it. Janine, are you there?
Yes, I'm here.
Okay, so just think of it that way. Your messenger RNA is always going to have the opposite complementary bases to what is on the DNA. And your transfer RNA will always have the same nitrogenous bases as the original DNA had. Does that make sense?
Yes.
You sure?
Yes.
Okay. Any questions?
No.
All right. So let's move on from there now. So now you know what you're looking at there. All right. Identify organelles A and B. You've already done it. So all you do is you are going to write organelle A would be the nucleus, or you could say nucleoplasm. It's both correct. But they say organelles. So sorry, you'd have to say the nucleus. And B would be the ribosome. Label. Janine, are you there?
Yes, hello.
Okay, yes, we're having issues with our telephone lines. All right. It's all these gremlins when it rains, you see. Label structure C, E, and F. We've done that. We've said C was your messenger RNA. E is your transfer RNA. And F is going to be the amino acids, all right? Which stage of protein synthesis is represented at 1? 1 here is going to be transcription. And I've already explained transcription to you. All right?
Yes.
But 2 is going to be translation. Yes. Now, what do you think translation is? Look at it.
Isn't it taking what's been transcribed?
Yes, you're translating it. So you're saying, okay, there's the codon, so that's the opposite to what I want, the amino acid that I want. The amino acid that you want is CCC, so that it's the same as what was on the DNA. Or TAA would be UAA, because remember, we're talking about RNA here. Okay? If we look at this D here, what would D be? Now, you can either go opposite to what is coded here on the messenger RNA, or you can just look at what the DNA was. So it's going to be C, A, G. Does that make sense, hun?
Yes.
Okay. Now, let's check. Guanine to cytosine, uracil to adenine, cytosine to guanine. So is it correct? Yes, it is. But your translation, you are translating, your transfer RNA translates what was on the DNA because it is the opposite to what the messenger RNA is telling it. Then it knows, okay, I must now go and find an amino acid that has that combination, or an amino acid that has that combination, or an amino acid that has this one. So that we can take our amino acids and we can string them together and group them together to make the protein that is specific for your body. All right?
Yeah.
Does it make sense?
Yes, sir.
Because I promise you, once you twig this, once you've got it, you don't have to learn a thing. It will be in your head till the day you die one day. Okay, you'll be an old granny and you'll be able to explain protein synthesis. It's just a matter of understanding transcription. Your messenger RNA comes into the DNA and it copies in the opposite what is on the DNA. It codes it. It then goes to the ribosome and it says, okay, I'm here. I need the opposite of this made. And your transfer RNA comes in with the with the anticodon, and that gives you the recipe for the specific amino acid. All right?
Yeah.
Write down the anticodon that reads from left to right at D. So we've already done it. From left to right, it's going to be CAG. Happy?
Yes.
Sure.
Yes.
Have a lekker afternoon, my hun.
Thank you.
All right.
Thank you.
Pleasure.
Bye.
Cheers. Bye. Okay, now tell me, my hun, or rather teenagers, you want male reproductive system?
Yes, ma'am.
Okay, so we've got we've got it on the board at the moment, the human male reproductive system. First thing you're going to have to do, my darling, is you've got to know what the labels are, okay?
Yes.
So first thing you are going to do is you're going to tell me what these labels are. So what is this one here? It is come.
Which one, ma'am?
A.
It's the prostate gland.
That's good. Okay. And what is B?
It's the vas deferens.
Good girl. And C?
The epididymis.
Epididymis. Okay.
Yes.
And D is the?
Testis.
No. It's the testis. It's one. Testes with an E means they're 2. Testis, 1. All right?
Okay, ma'am.
And E is the
Urethra.
urethra.
Yes.
And remember, the urethra thrusts the urine out. The ureters take the urine from the kidneys into the bladder. The urethra is from the bladder out of the body. All right?
Yes.
So that's how you remember the difference. Okay? Now, F is going to be the...
I don't know that part.
It's an acrosome. Look at it. A-C-R-O-S-O-M-E. It's the acrosome. And G is the nucleus. Okay. The acrosome is right in the front of the head of the sperm cell. This is a little sperm cell. Okay. And what it does is when it comes to the female egg cell, it releases enzymes, and those enzymes then open the cell membrane up so that the sperm cell can get in to the egg to fertilize the egg. Okay?
Okay.
All right. So that's the acrosome. That's what it's responsible for. Okay. So the first thing we have to do here is it says provide labels. Provide labels A, B, E, and G, which we've done. Okay? You see here? We've done them. But the reason you're not doing double, you're not writing everything out double, the reason for doing this is so that in your head you know what you're going to be asked. You understand?
Yes.
And you know what all the parts are. And then it's easy to just write it out properly. I mean, you can just put letters here or just half words because it's for you. It's not for anyone else. No one sees this. They're going to see what you write on your paper. So here you're going to say A is the prostate gland, B is the vas deferens, E is the urethra, and G is the nucleus. And it's the nucleus of the sperm cell, all right?
Yes.
Okay. Then state one function each for C and F respectively. Now, what does the epididymis do?
Sorry?
What does the epididymis do? Because C is the epididymis. What does it do?
It stores the sperm.
That's
temporarily
correct.
and
100%.
later.
No, you just have, that's all you have to say is the epididymis stores the sperm cells, okay, while they mature. And then F, the acrosome, I've already told you, what it does is it releases enzymes which break the wall, or I mean the cell membrane to the egg cell. Okay?
Okay, ma'am.
Happy?
Can you repeat that?
Okay, I'm going to write it here. F is the acrosome. Okay? And what does it do? It has enzymes are released which penetrate, or you can say break, the cell membrane of the egg cell. Okay? Or ovum. All right. So if I've got the little egg cell sitting there and it's got its own little nucleus, you've got the little sperm cell comes along, and in the front is the acrosome here, and it makes a little hole so that this little head can go in, and then the tail breaks off. You understand?
Yes, ma'am.
Okay, Mahan. Right, now, state the letter and the name of the part where the sperm are produced. The letter would be D. Okay, and the name, the testes, or testis. Testis is 1. Testes is if there are 2. All right?
Okay, ma'am.
Okay. Explain why it is necessary for part D to hang outside of the body of the male. Because sperm is produced at 35 degrees Celsius. And what is our body temperature?
Don't know.
37 degrees. So your normal body temperature is about 37 degrees or 37.2, 36.8, but it's about 37 degrees. So the fact that it's 37 degrees, the sperm cells are produced at about 34 to 35 degrees. That's the optimum temperature for little sperm cells to be produced.
So they must be kept at low temperature.
Slightly lower than the body. So that is why when it is very cold, the scrotum, which is the bag, will then pull the testes closer to the body so they can get warmer. And when it is very hot, the scrotum moves away from the body, and then the testicles are further away from the body so that they can cool down. You follow?
Yes.
Does that make sense?
Yes, ma'am.
Okay, my hun. All right. So sperm cells are produced at 35 degrees. Body temperature is 37 degrees Celsius. Therefore, they must move away to control temperature. Okay?
Okay, ma'am.
Does that make sense?
Yes, it
You
does.
understand that, though? All right,
Yes.
then. The cells that secrete the male sex hormone. Now, what cells secrete the male sex hormone? Do you know?
Yes.
What are they?
The cells of Leydig.
Right, good girl. Okay, the cells of Leydig. Then, and the cells of Sertoli, what do they do?
The cells of what? Ma'am?
Yeah?
The cells
Sertoli.
of what?
The cells of Sertoli, what do they do?
I think they produce the spermatozoa.
No, it's nutrition for the spermatozoids or spermatozoa. Okay.
Okay.
They are in the germinal epithelium. That's where they're found. And they produce the nutrients while the little sperm cells are being formed. So that you
Okay.
don't confuse cells of Leydig with the cells of Sertoli. All right. The hormone that stimulates the development of secondary sexual characteristics, it's what I think some guys have too much of. It is
Somatotrophic hormone.
testosterone. Here we go. Testosterone. Okay? And testosterone is what makes guys all, you know?
Okay. ma'am.
Okay? And it's their secondary male characteristics. Describe a vasectomy. During a vasectomy, part B is surgically cut. So let's just have a look here again. Where's part B? It's the vas deferens gets cut. All right. Now, if we look at it being cut, it says, explain how this procedure will act as a method of contraception. Because when you cut, remember, the testes are going to carry on producing the sperm cells. And the sperm cells then will go into the epididymis, and when they're ready and they mature, they're going to now move into the vas deferens. But the vas deferens is cut. It's like having a road with a bridge, and the bridge is smashed down. Nobody can travel across, can they? Because there's no road. So what happens is then the sperm cells are just reabsorbed. So these sperm cells can't get out because the road is blocked. It's like having this big block in the road. So they can't move out through the vas deferens. Does that make sense, hun?
Yes. ma'am.
Okay, so why is it a good method of contraception? It's almost 100% proof, is because the sperm cells cannot move beyond where the vasectomy was or where the cut is, so it cannot be ejaculated into a female. All right?
Yes.
Will it be possible for a man who is HIV positive to pass the HIV virus to another person if he undergoes a vasectomy? Now look at this.
Yes.
It's actually a very clever question. If I cut the vas deferens, there are going to be no sperm cells because that's where the sperm comes from. But remember, we have seminal fluid. And seminal fluid is made up from the Cowper's gland, it's made up from the vesicles, it's made up from the prostate. All of those glands are going to secrete fluid in which the sperm are transported. You understand?
Yes.
The HIV virus is going to be in there, and in there because it's in the fluid. So if a person's had a vasectomy, the sperm can't come out, so there's no more sperm, but those fluids from the prostate and the carpus and the seminal vesicles that makes up the semen, that is still going to be there. You follow? And that's what carries the HIV virus, not the sperm cells. You follow?
Yes.
So if we go back to our question here, is it possible for a man who is HIV positive to pass the HIV virus on after he's undergone a vasectomy? Yes. Yes, it is. Why? Because of the fluid of the seminal, or you can just say the seminal fluid. There are no sperm cells,
But semen is still released.
but HIV virus. You understand? Okay,
Yes,
explain
sir.
your answer. I've just explained it to you. But does that make sense? If I
Yes, it
cut that
does.
tube, there are no sperm cells. But what do sperm cells swim in? They swim in that fluid. And if they swim in that fluid, well, the fluid is still there. You follow?
Yes, I
When a
do.
male has had a vasectomy, he still ejaculates a fluid, but it's the fluid without the sperm cells. Does that make sense, my darling?
Yes, it does.
Okay, well, there we go. That's the end of your question. Yes?
Ma'am,
Mhm.
can I ask you something?
Yeah?
The essay type questions, how do I tackle them?
Your essay type question.
Yes.
First of all,
Do
you
I write in point form or in a paragraph form?
No. You don't write in point form. You write it in a paragraph type form. You do not put headings, etc. Okay, it must be like a paragraph. It must flow. All right. I'll tell
Okay.
you what.
I don't write subtopics. I just write a paragraph.
Yes, you mustn't put point form. You mustn't put bullets. You mustn't do diagrams. Just look at what they ask you. They normally ask you 2 or 3 things. And then just make sure that you answer each of those 2 or 3 things in a paragraph. All right? Each
Okay.
one with its own paragraph. What you can do in the paragraph is you can say, for example, contraception, and then explain contraception. Your next paragraph could be safety of a degree of prevention, and then you can carry on. But you follow what I'm saying. But don't do a heading and underline the heading. You put
Okay.
what you're answering with a colon, and then you answer your question. So it's easy for them to see and read. All right. But please,
Okay,
when
ma'am.
you do it, follow exactly what's asked in the question. And just say, right, that's point 1, that's point 2, that's point 3. And that's how you answer it. First paragraph, second paragraph, third paragraph. All right.
Okay.
It's just a 12-mark question. Don't think of it as an essay.
Okay.
Okay. All right, my
Thank you
darling,
very
good
much.
luck, hey? And just keep your
Thank
head
you.
about you. Read your questions carefully and only answer what is asked, okay? Next caller on the line, and I believe your name is Kakala. Are you there, Kakala? Okay, they're still busy trying to get him on the line. People, when you do get through and the studio does call you back, please switch your TV's sound down a little bit or quite a lot. Otherwise, there's a whole echo and a mess on this side. But let's have a look in the meantime while we're waiting for Kakala to come on. This is the question that was asked. Kakala, are you there?
Yes, I'm there.
Where are you?
I'm from Limpopo.
Are you from Limpopo province? That's nice. I'm going to be visiting your beautiful province in December on a holiday. And I'm actually very excited.
Oh, that's good.
Yeah. Okay, Kakala, this is your question. It's clearly on inheritance of characteristics such as blood groups and genetic disorders over a number of generations. What they've given you here, or the question that you've got, is they've given you a pedigree chart. Now, we're going to go through the question. It says the pedigree diagram below shows the blood groups of individuals of a family. So let's underline the blood groups, and let me get my pen back. The blood groups of individuals in a family, the blood groups are indicated inside the circles, okay, or the square. Now remember, a circle is generally female and the square is generally male, and I see it's exactly like that in this diagram. The blood groups of individual W and X are not indicated, so clearly they're going to want you to do that. All right? So, Kakala, what you need to do whenever you get a pedigree chart, you say thank you, thank you, thank you, thank you, go mwah, and then start answering your question. Because everything is given to you here so that you can work it out easily. All right, now, remember there are 3 types of blood groups. There is A, there is B, and there is O, and then there's AB. Why? Because we have co-dominance. Okay, dominance. In other words, you're going to find A and B equally dominate, but O is recessive. Okay.
Yeah.
So O, when it's in its heterozygous state, is going to, in its homozygous state, it's going to be completely recessive. It's a wuss. The only time a baby will have, or a person will have blood group O is if it is double O. All right, so look here.
Yeah.
So that is my recessive trait. And you always go to your pedigree chart and mark your recessive trait first. Okay, now, this one here is blood group O. It must be OO. All right? This one's blood group A and blood group B, which means these 2 people here had a child, okay, or 2 children. The one is blood group A and the other one is blood group B, but the mommy is OO.
Yeah.
If she had an A or a B gamete or gene sitting in here, she would have been that one. So in order for them to tell you her blood group is O, it means it must be homozygous recessive OO. Does that make sense?
Yeah.
You sure?
Yeah.
Kakala?
Yes.
Okay. But now, we've got a baby with an A and a baby with an O, which means this baby must be AO and BO. You with me?
Yeah.
Which means daddykins must be what? Where's the A and the B come from? It doesn't come from the mother, so it must come from the?
Father.
That's right. So the father must be AB.
Yeah.
Do you agree with me?
Yeah.
Sure?
Yeah, sure.
Okay. Now, here we've got A, O, and this baby here ends up being OO. So how are we going to work out what X is? Come, look here. What would X be? What would you say X is?
X is the mother, and I think she's...
What is she going to be?
She's going to be blood group B. Yeah.
No. She can be anything as long as there's an O. Because we have to take an O from the mother and the O from the father to make this baby blood group O. You understand?
Yeah.
So she can be OO or she can be AO, or she can be BO. We don't care. As long as there is an O there somewhere, because this child must have OO.
Yeah.
You follow?
Yeah.
And this one here can't be AA because the mother here was OO.
Yeah.
You follow?
Yeah.
Are you happy?
Yeah, I'm happy.
All righty. Now let's look at our questions. And it says, write down all the possible genotypes of the individuals, W and X. Now, W we've already established as AB.
Yeah.
Okay?
Yeah.
It can only be AB because this baby is A and that one is B. And the mother is OO. And here, X, what have we got? It can either be OO or BO or AO. As long as there's an O, because this child has to get an O from the mother. You with me here?
Yeah.
All righty. Now they say here, hemophilia is a blood clotting disorder. Okay?
Yeah.
And with that blood clotting disorder, explain why mainly males suffer from this disorder. Now that's very, very easy because hemophilia, the gene that causes hemophilia, sits on the female's X chromosome.
Yeah.
Okay, and if it sits on the female's X chromosome, the male Y chromosome doesn't have a compensating or an allele. You follow? It doesn't have a compensating allele. So what you would write here is the first point you would make is you would say that hemophilia is a sex-linked disease. Okay? By sex-linked, it's linked to the sex chromosomes or the gonosomes. You with me? So hemophilia is a sex-linked disease, disease number one. Number two, it is caused by a recessive allele.
Yeah.
All right. You with me here? And where's that recessive allele, which is carried on the X chromosome? You with me? It's carried on the X chromosome. All right. So now remember, in males, the gonosomes, which is the sex chromosomes, the chromosomes are XY. Yeah. Now you see here, if for example, and I'm just showing you this, but not there. Let's say the hemophilia sits there, the gene sits there. There is no complementary allele here because this doesn't exist on the male. Yeah. And remember, it is a recessive gene. So it can only come through if it's on its own. So there is no complementary allele here. So there's nothing to complement it or to cancel it out and make it heterozygous. You with me? Yes. Okay. So the male gonosomes are X. So I'm going to just take this out quickly. Chromosomes. Okay. Whereas your gonosomes are X, Y. All right. And then with your females, gonosomes are XX. So, yeah. So there is a dominant allele present. You with me? Yeah. So if it's sitting there and there, you've got this allele here, which will cancel it out. You with me? Yeah. And that is why only men get it. And many, many, many years ago, the Tsar of Russia and the Tsarine produced a child that had what was a hemophiliac. And the problem with hemophiliacs are they have no blood clotting ability. So they can just go boom and they bump themselves and they almost bleed to death. You follow?
Yeah.
And that's, you know, they made pop songs about Rasputin, and Rasputin was so-called doctor that kept this child alive. And he had some muties that he used to keep this child alive. But it's genetic, and it's carried across from mother. The mother is the carrier, all right? And the girls generally don't get it. It's the boys, because there is no compensating allele, because this is a recessive allele. You follow?
Yeah.
Okay. Anything else you want me to go through, hun?
Oh, you start off for question 3.1.1.
Sorry, you want?
3.1.1, is that all? For question 3.1.1.
Yeah, this is 3.1.1. You've got the pedigree chart, the blood groups. So the groups, the blood groups of individual X and W and X are not indicated. So following from that, they say write down all the possible genotypes for this. Now the mark allocation here would have been what? I think 3 marks, yeah, it would be 3 marks.
Yeah, but it carries 8 marks, and B.
Is it 8 marks? So write down all the possible genotypes. So let me think here. Your genotype there can only be AB, so that's 2 marks. Your mark for here, it can only be OO, which is 2 marks, BO, AO. So that's 2, 4, 6, 8. There's your 8 marks.
Okay.
That's all you needed. What else do you want to write, darling?
Oh, there's nothing else.
Hey, Kakala, have you had any other questions? Now look how easy that was. That is 8 marks for no work. Just remember, whenever you get a pedigree chart, work backwards.
Yeah.
Look at where the characteristic for two recessive genes sits.
Yeah.
And whatever that, so if they tell you a bent pinky is recessive, then you know it is little p.
Yeah.
You follow? Or little f, because it's the little one. Tongue rolling. If they tell you that keeping your tongue flat is recessive, then you put flat tongue. Wherever the flat tongue is indicated, you can see it from your pedigree chart. That's why you get a question with a pedigree chart, you rub your hands in glee and think, piece of cake. All right.
Yeah.
And work backwards. Go with what is recessive. And remember, just look at this here. If there is any recessive trait, let's say blonde or blue eyes versus brown eyes, if it is BB, okay, it's going to be recessive, which means I've got parent 1 and parent 2. There's the dad and here's the mom. Yeah. In order to produce a child that has little b, it means that either both mom and dad must have the little b present. Now, if mom or dad are little b, we can see it.
Yeah.
But both mom and dad can be heterozygous. Mm-hmm. But mom and dad cannot be homozygous dominant. Why? Because both of them have to give a little b.
Yeah.
So that's how you can always work out what the parents are based on what the children are. Does that make sense?
Yeah, it makes.
Okay, my darling, we're going to go for an ad break. Kakala, work hard, put in the effort, don't study tomorrow morning and don't study till late tonight. Get to bed early enough to have a good sleep. Tomorrow you have a good protein breakfast, okay?
Yeah.
All right. Have a boiled egg if you don't like eating, or yogurt. But eat protein, go to school, don't talk to anyone. If they come with, did you learn this? Did you learn that? Because all they do is confuse you. Tell them to go away. Yeah. Breathe in the good, out with the bad. Read your questions and then answer them. And think before you ink, okay?
Yeah.
All right. We're going to an ad break. Hi guys, we're back again. I've been told I've got Amobella. Are you there, darling? Okay, while we wait, Amobella.
Hello.
Good evening, and how are you?
Good, and yourself?
Are you
Just a
working?
bit nervous before.
Why?
The exam for tomorrow.
No. Listen to me, darling. Don't ever be nervous for an exam. Okay? Don't ever be nervous for an exam, because you know what nerves do? Nerves help you not to think clearly. Walk in there and say, just give it to me. You know, if you're going to play a soccer game or a rugby game or a cricket game, so that it's good to have a little bit of butterflies and all the rest of it and be excited. But it doesn't help to go in there nervous, because you'll get bowled out to the first ball and then you walk the golden duck. You follow? You're not going to do that. You're not going to do that. You're going to prepare properly. You're going to go to bed early. You're going, or early-ish. You're going to get at least 6 to 8 hours of good sleep. You're going to get up tomorrow morning ready to rock and roll. You've been studying for 12 years. 12 years. It doesn't help getting all nervous for a 2-hour exam or a 2.5-hour exam after studying for 12 years, hey?
That's true,
Now,
eh?
Amobella, you've got meiosis here, all right? Let's go to your question. And it's actually easy. Just keep your head about you. Okay, remember, there's interphase, which you can see nothing. Then there's prophase, metaphase, anaphase, and telophase. Now, interphase, you can see nothing. Metaphase, everything's always in the middle. So that's easy to pick up, okay? And if it's in little X shapes, and I know I sound ridiculous, but this is the best way to remember it. If they are little X's, then it's meiosis 1. If they're little V's, it's meiosis 2. It's as simple as that.
True.
Yeah, look here. If they, you see what they look like here? They look like little V's. Do you see that?
I can see.
Okay. Now in meiosis 1, they're going to be little X's. In meiosis 2, they're little V's.
Okay.
And that's how you tell the difference.
Oh.
Easy, hey?
Yeah, that's pretty easy.
Not very scientific, but it's easy to remember it that way. Okay, so that's number 1. Then, so metaphase, everything's in the middle. Telophase, ach man, please, everything's sitting, separating. It looks like a little 8. Or it's a square, a rectangle with little lines coming across if it's in plants. But in animals, you're going to have your invagination, so it goes into the shape of an 8. So it's easy to see. All right. Now, the trick is anaphase versus prophase. Now, prophase, everything will be mushed, but you can see your chromosomes, and during prophase 1, you're going to have your chiasma, which causes the crossing over. And that's for genetic variation. Okay. It's as easy as that.
Yes.
So they are there. They're together. They're close by, the chromosomes. And in anaphase, well, remember, anaphase follows on from metaphase, so they're all in the middle and nicely lined up. And anaphase, they're starting to pull to the poles. And that's how you recognize it. Okay, so if we look at this here, it's anaphase 2. Why? Because we've got the V shape. When they ask you here what is A is a chromosome. So we're going to write that. But your chromosome is going to be haploid. All right. And here, B is your spindle fiber.
Oh.
Okay? These are your
Spindle fibers,
poles.
yes.
This is where the equator was, down the middle. It's not difficult. Look at that. Simple, simple. All right. So it says provide labels for A and B. Well, we've already done that. Okay? Then it says, so this will be your mind you, it's be your chromatids. Sorry. So it's a chromosome, but they're haploid, so it's got to be chromatids. So we're going to put here our daughter, let's make it correct, our daughter chromatids, yeah. Okay, daughter chromatids. That's what you're going to find there. Or your daughter chromosome you can put. But it's the daughter chromosome that's important. Daughter chromatids, daughter chromosome. All right, so that's A and B. What phase of meiosis? Well, it's anaphase.
2.
And what have we got? We've got the V's, so therefore it's anaphase 2.
2.
Hey, it's simple. How many chromosomes will there be in each cell formed from this cell at the end of meiosis? So they want to know how many chromosomes will there be in each cell. So tell me, how many chromosomes can you see here?
I think there will be 2.
Yeah, 1, 2, 1, 2. All right, so 2.
Okay.
How many chromosomes are present in the phase before the one represented in this diagram? There were
2 as well.
Yeah. Okay. How's this hard?
Okay.
Hey?
No, just the sketch confuses me a bit, eh?
Okay, Amabella, look here. Let's do it this way. Okay, look here. We have interphase. They're not going to give you interphase because it's a nothing phase. Everything that happens in interphase is chemical. It's chemical. Can you see chemicals? Can you see chemicals? No, you can't, darling. If I take a glass and I chuck a couple of chemicals in, you can see nothing. So when you look at a cell that's in interphase, you physically cannot see anything happening. All right. So we're going to work on the other 4 phases. So we have interphase. Then you have prophase. Then you have metaphase. Meta means middle. Okay? Anaphase.
Anaphase. Telo as well.
Telo or telophase. If it's American, it's tela. And if it's anywhere else, normally your English textbooks, your UK ones are telophase. So you can write either telophase or telophase, same thing. All right. Now, if you look here, with prophase, okay, remember everything you end up with your cell membrane because everything else just moves out of the way. But you're still going to be able to see, the nucleolus will have disappeared. You're going to have your cell membrane will still be around. In prophase 1, not prophase 2. And in here, you're going to have your chromosomes, and they are going to be crossing over. Okay? So you're going to be seeing crossing over. So that should be prophase 1. In prophase 2, they're going to be sitting there like this. But you're going to see where they've crossed over.
Oh.
And that's how you're going to know that you're going to still be able to see your pieces. All right. Metaphase, you're going to have everything sitting here. Okay? Centromeres, centrioles, your spindles, etc. Okay? Anaphase, so meta, just think middle. Ana, they're starting to pull away. They're going to the poles, so they're pulling away. You follow? Okay, I'm just drawing them. Okay, those all had 2 and this one's got 3. Don't ignore that. What I'm trying to show you is that they're pulling towards the poles. You follow? Telophase.
Yes, I follow. Where does karyokinesis fall under this and cytokinesis?
Okay. not you've got karyokinesis and cytokinesis. Cytokinesis is going to be during telophase. This is your cytokinesis. Okay? Cytokinesis is the splitting of the cytoplasm. So where is the cytoplasm going to split? Only during telophase, here, when it does this. Now this is Sept, this invagination gets bigger and bigger and bigger until it separates into your 4 identical daughter cells.
Oh.
You understand?
Yes, I understand quite clearly.
So it's here. This is when your cytoplasm starts to separate during telophase. Your karyokinesis. This is the splitting of the, I'm just going to put split of nucleus. Now, where would the nucleus split here? It is during metaphase. So you have metaphase, early metaphase, but during metaphase, as soon as you go into late metaphase, karyokinesis has taken place. So karyokinesis takes place during metaphase. You're splitting the nucleus. Makes sense?
Yes, it does.
Amabella, don't get hooked up on little stupid detail things. Okay? Know the big picture.
Okay.
Know how to identify your different phases of meiosis. Make sure that you can identify meiosis 1 and meiosis 2. Without a doubt, your most important phases are anaphase 2, prophase 1. Let me do them in order. Prophase 1, metaphase 2, and anaphase 2. Those are your most important ones because that's when things happen. Okay?
Okay, thank you very much.
Okay, perfect, darling. You just don't be nervous. Make sure you get a good night's sleep. And if you can't sleep, go and get a glass of milk and put a whole dollop of honey in there or 3 or 4 spoons of sugar. Okay? And I promise you now, you will go do time so quickly your head will be spinning. Okay? But you need a good night's sleep. Do not attempt to study till 2, 3 o'clock. Do not get up at 4 o'clock in the morning to study. If you don't know it by tonight, 10:30, 11, the chances are you're never going to know it. What you do, though, is when you get up and you study early in the morning, your brain is so busy trying to remember what you studied early in the morning just before you write that it forgets everything you've learned for the last 11, 12 years. You follow? Don't do that to yourself. Maybe read through summaries just to get your butterfly settled a bit, but don't sit and swat early tomorrow morning. Get a good night's sleep, have a good breakfast, and go in and write your exam with confidence. Read your questions properly and make sure that you can graph, make sure that you can calculate. I mean, they're very silly calculations. They really aren't difficult. Make sure you're able to calculate and make sure that you can draw a table of results from a graph, and you'll be fine. Okay?
Oh, thanks a lot.
You're going to get a distinction tomorrow.
I'm going to aim for it.
There we go. That's the spirit. Not, ooh, I'm nervous. No, you're not. You're going to slay that dragon. Okay? All right, there we go. Amabella, good luck, my darling. And we're going to, for the rest of you, we're going to go to the studio for a quick ad break, and I'll see you all in a minute. Okay, guys and girls, we back. Ntabi's on the line, I hope. Ntabi, are you there?
Yes, I am.
Where are you calling from, Sweet Pea?
I'm calling from Mpumalanga.
From a Pumal, still my favorite, but okay. That and Durban, I don't know. I'm a Shock supporter, so I'm very pro Durban as well. Okay, my hun, your question here is question 3.4. It says the investigation was conducted by grade 12 learners on fruit flies. So now we are going to underline fruit flies so that we remember that these stupid little animals, and what do they have? They've got red eyes and white eyes. By now you should know that, okay? And we're looking at the genotypic and phenotypic ratios. Okay. Now, Ntabi, what does genotype mean?
It's the genetic makeup of an organism.
Right. It's the can you see it?
Yes.
Sometimes. You can only see the genotypic.
Oh, no.
You can only see the genotype when it is expressed in the phenotype, and it can only be expressed in the phenotype if they are dominant and recessive. It's very specific dominant and recessive genes, okay? And if we have F1, that's your first filial generation, so it's the first lot of babies. F2 is the second filial generation, which is the second lot of babies, okay?
Okay.
All right. They placed 12 homozygous red-eyed females in a flask and 12 homozygous white-eyed males in the flask. The flask contained ripe fruit, clearly, so we want to give them food so they don't all die, because that's what fruit flies do. The opening of the flask was covered with cotton wool. Why is it covered with cotton wool? Come think. Why would it be covered with cotton wool?
I don't know.
Think. If I've got a glass of water here. If I take this glass, and let's say I put the fruit flies in here, okay, without the water, but I'm in a glass. How do I stop them from flying out? I must cover it, mustn't I?
Sorry?
I must cover it.
Yeah.
Otherwise, they're going to fly out, so I must cover it.
Oh,
But
yes,
if I cover it with something that's a seal, like a plastic cover, then what's going to happen? Those fruit flies are going to run out of oxygen.
Yeah.
And then they'll die.
Yes.
So the cotton wool has two functions. One, it's going to stop the fruit flies from flying out. But at the same time, it's going to allow gases to go through because the gases can move through the cotton wool.
Yeah.
So they don't all suffocate. You happy with that?
Okay, yes, ma'am.
Okay, and if you look at the first question, it says, give 2 reasons why cotton wool is used. 1, to prevent the fruit flies from escaping. Number 2, so that gases can exchange. Otherwise, our fruit flies will suffocate. Okay. Does it make sense?
Yeah, it does.
Okay. You see, that's an easy question if you just think a little bit. Okay. After 3 weeks, the F1 offspring were crossed, and the results are F2. Now, you know what? I want to show all of you this. There are only ever in the life science that you do, there are only 4 crosses. There are only ever 4 possible crosses. Okay? Okay. Ever.
Okay.
So write these down. Okay. Cross number 1. Okay? Cross number 1. I have a homozygous, so I'm going to have a homo and a homo. So I'm going to have homo dominant and a homo recessive. Okay? So let's go with brown eyes and blue eyes. So for all of them, we're going to say B is brown, it's dominant, and small b is blue. Okay? It's recessive. I've written it with a small letter. Okay? So homozygous dominant parent, we're going to have capital B, and homozygous recessive, little b, little b. All the offspring are going to be the same. They are all ever going to be heterozygous. You see that?
Yes, ma'am, I do.
Okay, so we're going to have 4 to 4, capital B, small b, heterozygous, brown. You with me?
Yes, I am.
Number 2, we're going to have homozygous dominant, and we're going to have heterozygous. Okay, so homozygous dominant and heterozygous. I'm going to have capital B, capital B, capital B, capital B, capital B, small b, capital B, small b. In this case, I'm going to have 4 in 4 or 100%
Uh-huh.
are going to be brown.
Yes.
Okay, that's my
Yes.
phenotype. But that's my phenotype. But
Yes.
for my genotype, what am I going to have? I'm going to have 50% are going to be capital B, in other words, homozygous brown, and 50% are going to be heterozygous brown. You with me?
Small b. Yes, ma'am.
Happy? Okay.
Yes.
The next one, number 3. I'm going to have heterozygous plus heterozygous. Okay.
Okay. Okay.
And they often, this heterozygous is asked 90% of the time. They either ask homozygous dominant, homozygous recessive, or heterozygous and heterozygous.
Okay.
It's easy. Look at this. Heterozygous, capital B, small b, capital B, small b. That's going to give me capital B, capital B, capital B, small b, capital B, small b, small b, small b. It is always a 1:2:1 ratio.
Yeah.
You see?
Yes,
But
ma'am.
3 to 4 are going to have the dominant 3 out of 4, or 75%,
Yeah.
are going to be brown
Brown.
because of the dominant gene.
Okay.
And 1 to 4, or 25%,
5%. Yes.
are going to be blue.
Blue.
But it's always a 1:2:1 ratio if it is heterozygous plus heterozygous.
Okay.
Okay. And your
Okay.
last one is very similar to number 2, where I'm now going to have homozygous recessive plus heterozygous.
Okay.
So I'll have homozygous recessive little b, and capital B, small b, and it's pretty much the same kind of ratio that we had before, but except that I'm now using the recessive trait, 50-50. You see that?
Yes, ma'am, I do.
So 50% in the phenotype will be brown, 50% will be blue. In the genotype, 50% are going to be heterozygous and 50% are going to be homozygous dominant. Homozygous recessive. You happy?
Yeah, I'm happy.
Now, those are the only 4 crosses you are ever going to see. So if you know them, you know them. You follow? Okay.
Yeah.
So
Yes, ma'am.
the F1 are all going to be heterozygous. Okay? So if we use capital R and small r, they're going to be capital R, small r, capital R, small r, capital R, small r, and capital R, small r. The whole bang shoot, all the offspring. Why?
Okay.
Because both the parents were homozygous. You see, homozygous red eyes, homozygous white. So we've got
White.
we got RR. What am I doing here? Hang on. Let's do this. Let's go. Capital R, small r, small r. All the offspring are going to be capital R, small r. You see that?
Yes, ma'am.
In
I
the
do.
F1.
Okay.
Now we take our F2 and we are going to cross them. So what have we got? We've got heterozygous.
Heterozygous, yeah.
So we've got capital R, small r, capital R, small r.
Small r.
There we go. Does it make sense?
Yes, it does.
Now what have I got? I've got a 3:1.
Ratio.
Yeah. Or 3 and 1. So that's 75%. And this is 25%. So it's 3:4 and 1:4. You with me? So
Yes, ma'am.
give a reason why the cotton wool we've done. State one way in which the reliability of the results could be improved. Now, listen to me carefully. Whenever they ask you, how can you improve any kind of investigation? Okay, you are going to always say you repeat the experiment again and again.
Okay.
The more times you repeat it, the more results you've got. You then get an average of those results. So if there's a little change here and a little change there, you're going to have accurate, better results. Do you understand?
Yes,
So
I do.
whenever they say, how do you improve an experiment? You repeat it more than once, more than twice, more than 10 times, but you repeat it. Okay?
Okay.
Number
Okay, ma'am.
2, make sure that you have this, okay, here they have the same number of males and females, but sometimes in experiments, you won't have the same number of males and females, or blacks and whites, or pinks and purples. So you will always say, and I'm just going through all the different types of experiments, you will say that make sure that there's the two things you're comparing, that you have the same number of each. Okay. If it's to do with flowers or animals or plants, you're going to say that they have come from the same species.
Okay.
So you must use the same species of plant or animal when you're doing it. I'm trying to think what else. You would, I can't think of any more. You'd do it often. You'd repeat the experiment. You would you'd make sure that whatever you're doing is accurate.
Yeah.
So all your counting must be very accurate.
Okay.
And your sample size must be big. So for this specific question, you say you'd repeat the experiment, and that you would make sure that you can increase the number of fruit flies you do. So if I have 150 fruit flies, the chances are my results are going to be more accurate. You follow?
Okay, ma'am.
Does that make sense, darling?
Yes, it does.
Okay, so that is how you're going to improve the reliability. Repeat it over and over. Make sure that you're using the same species. Make sure that you're using more, so you select more, so you have a bigger sample. You can even do it with other organisms to see if it's the same result. Now, using the symbols capital R and small r to represent the alleles for eye color, state all the possible genotypes of the red-eyed fruit fly in the F2 and the white-eyed fruit fly in the... Now, the red-eyed fruit fly in the offspring, we've done here. If they're going to have red eyes, they can either be capital R, in other words, homozygous for red eye, or they can be capital R, small r, in other words, heterozygous for red eye. And the white-eyed ones can only ever be, because it's your two homozygous, your homozygous recessive, they must be little r. Makes sense?
Yes, ma'am.
Are you happy?
I am very happy.
Okay, good. Now, there was another question, or is that your question? That's it?
It was 3.3.
Was it 3.3? It's the one about this one here. Read the passage below and answer the questions that follow. Vaccine-producing bananas.
Yes, ma'am.
Okay. Now, if we go vaccine-producing, I'm just going to read this quickly, and then we're going to go for an ad break. All right.
Okay,
vaccines protect people all over the world. Now, we know we need vaccines. We've got to have vaccines for polio and measles and mumps, etc. Now, what these guys are saying is, they're working on producing genetically modified bananas that will act as a vaccine against hepatitis B, which is an absolutely awful disease. I mean, it's terrible to get it. It's a virus, and it makes you very, very ill. And it's a liver disease. It causes liver disease. Okay, that affects more than 2 billion people around the world. And they're saying in future, many vaccines and other medicines may be given to people and animals in an easy-to-eat fruit or vegetable. Now, I mean, this sounds so amazing. To me, it sounds like, you know, when you look at children and they don't want to eat vegetables, or they don't want to eat all these vitamins, and you don't want to pile your kids with vitamins, you can give them a banana or an apple and they'll eat it, and they're getting their vaccines instead of taking them for injections. All right, now it says here, look at this. State 3 possible advantages of using bananas as a vaccine against disease. Now think about what the advantage would be. Come on.
It will prevent the
Come,
body
darling. What would the advantage be? If you're feeding, first of all, bananas are delicious. Okay?
Yeah.
So it's a very delicious fruit. I'm not going to write this down. All right, I'm going to take too long. It's a delicious fruit.
Yeah.
Number one. Number two, it can be grown on large scale. Number three, it's easily accessible. Okay, you follow. So, I mean, there's already 3 possible answers that you could come up with. But bananas are if it's sitting in that fruit, that's great. Children will enjoy it. It's easy to give to children. You could give it to old people. You could give it to animals. You follow?
Yeah.
it's easy to grow. It's inexpensive because you can grow them on large scale. So anything like that, just use your common sense. Why would it be an easy way of giving vaccines? The advantage is that you're giving a fruit which is nutritious with other nutrients as well, instead of getting injections which are sore. You follow? Okay,
Yes,
3 reasons why people might be against the use of genetically modified. Well, first of all, there's a whole group of people out there that say any genetically modified food is bad. And genetically modified animals, it's bad. It goes against their cultural beliefs or their religious beliefs. They say it's wrong. Okay? Another reason could be because if you're using fruit or bananas, the shelf life is not very long. You know, if your vaccines, once they've made them in the chemical form, they can keep them for a year. Whereas if it's a banana, it's only going to last for a week or 2, and then the thing's going to lose its shelf life and start going rotten.
Okay.
So you've got a reduced shelf life. Maybe there are people that are allergic to bananas, so they won't be able to eat it. Maybe there are people that don't like bananas.
Yes,
so
I do.
they're not going to eat it. You follow?
Yes, ma'am.
There's also people will then maybe sell bananas for a lot more than the price now. So they'll increase the price so they're greedy so they can make more money because you're getting your vaccine in that banana. So they then they make it so expensive that only the rich people can afford it. But
Yes.
those are the things that you needed to come up with here. It wasn't anything you had to learn. You just had to think it through. But you had to understand what genetically modified food is. You follow?
Okay. Yes, ma'am,
Okay,
I
my
do.
darling, I'm going to say goodbye to you. We're going to go for an ad break, and we'll take the next caller. So good luck, hey? Work hard, and a nice 5-leaf clover just for you so you can do well. Okay, people, we've had about 50 callers and emails, etc., about plant reproduction. And it's such an easy little piece of section to do. So what we've done is we've actually loaded on a question from one of the old papers on plant reproduction. And we'll sort of go through this. It gives me the diagrams that I need to work with and to show you the question. We've got a caller that's coming on. I think her name's Walile. Walile, are you there? Okay, she's still coming on. While we wait for our caller, we're going to start with this question. So, Walile, are you there?
Yes,
Hello, darling. Where are you calling from?
Durban.
You're from Durbs. Now, listen, when you finish to this whole session, okay, at 7:30, you're going to go outside. You're going to go stand at your door. You're going to face the sea, and you're going to go, mwah. Kathy sends her love, and yay for the sharkies, okay?
Okay.
Good. All right, here we go. An investigation was done to determine the role of petals in insect pollination. Now remember, insect pollination or plant pollination, a flower has to get pollen because the male gametes sit in the pollen. The female gamete sits in the ovary. So the pollen the flower can either be self-fertilized, so its own pollen fertilizes its own egg cells, or
Yes.
cross-pollination must take place. Now, if cross-pollination takes place, it means that something has to get the pollen from the one flower to the next flower. You follow?
Yes.
Does that make sense?
Yes, it does.
All right. So now, determine the role of petals in insect pollination in apple flowers. Now, have you ever smelt apple flowers?
No.
They are beautiful, beautiful. They smell so pretty. Okay, now,
Okay.
so there's a lot of scent that goes out from those flowers, and the little petals are sort of white and very light pink. They're very pretty. When the flowers are self-pollinated, the pollen tube grows a little into the stigma and style, and fertilization does not take place. You see that? If it is
Yes.
self-pollinated, why? These plants require cross-pollination. So what these people did was they put together this experiment. They said, right, there are 10 flowers with petals and 10 without petals. And after 2 days, the flowers are prevented from further pollination. And then after 7 days, they looked at how far the pollen tube had grown down into the stigma and whether fertilization had taken place. Okay? So first thing they're going to do now, they give you all this information. They tell you there were 10 flowers with petals and 10 without petals. And this is what they look like. Here are your flowers with petals. Very pretty. And here are your flowers without petals. Now remember, if a flower gets pollen, okay, and the pollen does land on the stigma, and it has no petals, there's nothing to protect that pollen from being blown away as well. So that's just the thought. So just think about that. Okay. Do you know all the parts of a flower?
Yes, I do.
Okay, because it's easy. Sepals and petals, and they tell you stigma, the style, the ovary, and then you have your stamens are made up of the filament and the anther. And the anther is here. That's where your little pollen grains are. Okay.
Yes.
Now, the results are shown in the table below. So here's our table. There are flowers with petals. The pollen on the stigma, there's plenty. The pollen tubes in the style, a lot of them grow down. How many ovules get fertilized? Only 38. So only 38 make it in through the micropyle and to fertilize those little ovules, okay? The flowers without petals, oh shame, look. They have the pollen on the stigma, there were only 25, okay? The pollen tubes in the style, only 8 make it. And fertilized, only 4 end up being fertilized. You follow? Now, we could argue there that the pollen was blown off. We could argue that the pollen came from its own stigma, etc., because there's nothing to attract the insects. So there are lots of reasons why this little flower without petals has so little pollen on it. Okay, so now, give an explanation of the presence of more pollen on the stigmas of the flowers with petals than on the flowers without petals. So let's think of 2 explanations. One, they were, first of all, the insects were attracted by the pretty color of the petals. Number 2, the wind didn't, it was then protected, so the wind didn't just blow it all away. You can also, I don't know, those are the 2 I can think of. Can you think of anything else?
Yeah, probably the pollen, the sexual reproduction pollen. It has spikes which stick to the style, unlike wind pollinated plants.
Okay.
Smooth pollen.
You could also say that the more pollen in the stigmas with flowers than without. We can also say that the flowers without petals didn't attract insects. So what do we have? Self-pollination occurring. You follow? But those are all explanations that you could have given. All right, then, and that's just 2 marks. It's quick and easy. Explain why there are more pollen tubes present in the styles of both types of flowers than the number of ovules fertilized. So why are there more pollen tubes? I mean, here we've got 86. Here we've got 8. And only 38 and 4. So actually, if you look at the ratio here, your ratio of flowers without petals, 8 pollen tubes and 4 egg cells are fertilized, whereas here You've got 86 and only 38. So this actually had a better percentage rate of fertilization than we have with petal flowers, actually. Tell me if you really want to get technical. Now, explain why there are more pollen tubes present. Come, why do you think there are more pollen tubes present?
Pollen tubes because it has petals, the flower has petals.
Mm?
Yeah, and more insects are attracted to that flower.
It's also, remember that with this whole process, you've got your stigma, your style, and then your ovary, okay? Your pollen tube is going to grow down. Now, first of all, yes, it's got to grow down. Now, if you've got a whole bunch of pollen tubes growing here, some of them are not going to make it into that micropyle because in the middle here, all right, you've got your micropyle over here. So that's where that little whole thing is. So it's now got to grow, the micropyle's the hole. It's got to grow all the way around and in here. Before any kind of fertilization can occur. So there only a certain number of those pollen tubes can actually get to the micropyle and get in through the micropyle. You follow? There's only space for a certain amount to go through there. So that's also an explanation. You could also turn around and say some of the pollen tubes may be dysfunctional. You could say that there's more pollen tubes will develop than actually cause fertilization to ensure fertilization actually occurs. It's like sperm cells in humans. We've got 90 million sperm cells and only one is going to enter the egg cell. You follow? So it's to ensure that fertilization actually does occur. But that was 2 marks. Then state 3 ways in which this investigation could be improved. Now, we've gone through this. I went through it with my previous caller. They always ask you, how can you improve this investigation? So number 1, if you look at what you were given, you're given flowers, little apple flowers, only 10 flowers. Okay,
you can increase the number of flowers.
Exactly. You can increase the number of flowers. You can make sure that all the flowers are on the same apple plant or apple tree. So you're going to have 100 apple blossoms with petals and 100 apple blossoms with, and you pull the petals off. You follow?
Can they give a few amounts of light?
Exactly. So anything to increase the sample size. If you the same types of flowers, so use the same species of flower, or you could use the same flowers on the same tree. You follow? So that you make sure that it's the same. You would ensure that that the pollen was able to move around. So I mean that the pollen was actually free, that the anthers were now secreting pollen. You'd make sure that you are that your results are accurate, so that you've counted correctly. So all of these things, I mean, there are a whole bunch of things that you could do here, but they only wanted 3. Let's think what else you could have done. You could have allowed more days for pollination, because remember here, they only allowed 5 days or 7 days after pollination. They wanted to check the fertilization. Maybe allow for 10 days so that the odd little pollen tubes can actually get there. You understand? So you can increase the time, increase the number, make sure it's the same species, make sure that it's the same plant.
Yes.
All right. Does that make sense?
Yes, it does.
Perfect. Then we're done. Anything else you want me to go through?
Not really, because I understand now.
Okay.
Sorry. If you don't mind, can you please just explain asexual reproduction?
Asexual reproduction.
Yes,
okay.
Stolen.
Asexual reproduction occurs mainly by mitosis.
Mitosis.
Mitosis is the duplication of a cell. Okay? So you are going to have mitosis occurring. You can have it from, for example, cuttings. So there are a lot of plants that if you take just a piece of the root or a piece of the stem or a piece or a leaf and you plant it in the ground, it sprouts roots and it starts to grow. So those would be
Okay.
your cuttings that you could use. Vegetative reproduction, you have bulbs, and the bulb will make a baby bulb on the side of it. And once that bulb is able to live on its own, or it can be independent, it would be able to break off, and it would then, or you would separate it. If you look at budding, for example, in potatoes. Potatoes
Okay.
are tubers. So have you ever gone to a bag, if you've bought a bag of potatoes, or your mom's bought a bag of potatoes, and they're in a nice, dark, warm place, and after a while, when you get to the bottom of the bag, the potatoes are almost a bit spongy-ish, and they start to go a bit green, and they get these little growths on them.
Yes, from
each-
the auxiliary bud.
Yes, those are little buds that form. And it's called an eye. And if you cut, or that's called the potato eye. And if you cut a piece of potato around it with that little eye, and you stick it in the ground, you plant it, it grows. By the way,
okay.
you know how to stop that. How's this for indigenous knowledge from passed down from granny to granny, from great-grandmother to grandmother, etc., is you take an apple and you put an apple in your bag of potatoes. And it takes a long time for those potatoes to start budding. But that would be another way is budding. Runners is another way. Strawberries work with runners. Plant, and you have this little shoot that comes off it, and where it lands, it makes a new little plant. So you have runners. Okay? So that's vegetative. You don't need sex cells and fertilization, so meiosis fertilization to reproduce. It reproduces by pieces of the plant growing in a certain way. All right.
Okay.
Okay, my darling, I have to bid you farewell. Study hard. Good luck for tomorrow, and we're going to an ad break. Okay, I've been told I've got Caroline coming onto the line, and this is the question Caroline wanted, and I love this question because it shows all the different types of, it's got the hormones, it's got estrogen and progesterone, follicle stimulating hormone, luteinizing hormone, the thickness of the ovarian, at least the uterine wall. It's a beautiful, beautiful question. Caroline, are you there?
Yes, I'm here.
It's talking for 3 hours. Caroline, where are you calling from, Sweet Pea?
I'm calling from Limpopo.
From Limpopo, so we've had Mpumalanga, we've had Limpopo, we've had Gauteng, we've had KZN. You see, Western Cape they're so laid back, they're sitting with their mountain and waiting for everything to happen. Hey, Caroline. Okay, Caroline, it says here, the graph below shows the menstrual cycle, all right?
Yep.
The menstrual cycle and the influence of the different hormones. Okay, so before we do anything, let's look at our graph. Okay, we've got the pituitary gland, and what does the pituitary gland do? It releases 2 hormones. It releases the follicle stimulating hormone and it releases the luteinizing hormone. Okay, so either FHS, which is follicle stimulating, or luteinizing hormone, LH. Okay. Now, please, you've got to remember, follicle stimulating hormone will release, it causes the release of estrogen. And estrogen is going to make sure that the lining of the uterus is nice and thick and glandular and that there's a good blood supply so that if fertilization occurs, then the egg cell or the fertilized zygote can attach into that endometrium wall and develop into a fetus and an embryo. You with me?
Yes, I'm here.
Okay. So that's the function. Estrogen makes sure that that lining builds nicely. Progesterone makes sure that that lining stays there.
Sorry, it's what?
That the progesterone makes sure that the lining stays thick in case the person is pregnant. In other words, in case pregnancy results in fertilization has taken place. Okay. So, follicle stimulating hormone causes the release of estrogen. Okay?
Okay.
Luteinizing hormone is going to make sure that progesterone is released.
Oh, all right.
You follow? Those are one of the functions of those 2 hormones. Okay, so your follicle stimulating hormone is going to ensure that the follicle, now if you look at this diagram, that the follicle develops. This is going to be your primary follicle. Okay, so your primary follicle develops there. It develops into a secondary follicle. All right, this is the Graafian follicle. That's when it's ready. Here the Graafian follicle bursts and your little egg cell is released. All right. And then it forms the corpus luteum. When pregnancy is not taking place and there has been no fertilization, then the corpus luteum is going to degenerate. Okay? As it degenerates, so your progesterone level is going to drop, your estrogen level is going to drop, and that is what causes menstruation, which is here for the first 5 days. So generally from day 0 to 5, you are going to find here that that's when menstruation occurs. All right? Then the lining starts to build up on from day 6 and 7, and we then end up with our lining perfect here so that implantation can occur. You with me?
Okay.
All righty. So let's look at our, and all of these are lined up. You see that? Okay. Now, on which day does ovulation take place? Now, all you have to do is use your eyes. When do you think ovulation takes place here?
I think on day 14, yeah.
Exactly. On day 14, look, your luteinizing hormone is at a peak here. Your little corpus luteum, the Graafian follicle bursts and the egg is released. Your estrogen level is starting to come down. Your progesterone level is increasing. The uterine wall is nice and thick. Yay, hooray, it is going to be day 14.
Okay, yeah.
Or you can write the 14th day. But don't forget the day. All right, between which days does menstruation take place? Now, menstruation here takes place approximately day 0 to day 5.
Day 7.
No, why day 7? It's got to stop and then the lining starts to build again. So I would say day 0 to 5 or day 0 to 6.
Okay.
All right.
Yes.
Then state any one function. Nobody menstruates for 7 days. Do they?
Yes, I'm here.
No, Caroline. It's Caroline, hey?
Yes, yes, I'm here.
Okay, listen. No woman, or maybe 1 or 2 women out there, but most women menstruate for how many days? 5.
Okay.
Eh? Are you with me?
Yes, I'm here.
That's normal. It's 5 days.
Mm-hmm.
So
yes,
it's approximately 5 to 6 days. Period.
Yeah, it is.
All right.
Yes.
Now, state any one function of luteinizing hormone. Now, look here. You don't have to learn this. Look here. What do you think the function of luteinizing hormone is?
I think it's for the follicle to release the ovum or something. I don't know.
Exactly. It's the luteinizing hormone so that it can prepare and produce and cause the production of the what? The corpus luteum. That's its most important function. Okay.
Okay.
Secondly, your luteinizing hormone will cause the release of the egg cell. So it causes ovulation. So take your pick. Either one would have given you your marks there. All right?
Okay.
Describe the changes in the level of luteinizing hormone showed in the graph. Now, all they want is the changes in the level of luteinizing hormone. There's still nothing you need to learn here. Can you see?
Yes,
You've just got to be able to read here. So we look at the luteinizing hormone. Now, describe. What do you see here? We've got a constant amount of luteinizing hormone for the first 14 days.
Yes.
Okay, or in fact, we can say here, if that's 7, so that's 7. So 8, 9, 10, 11, 12, 13, 14. Okay, my lines are a little bit messed, but okay, let's say, so we'll say approximately round about day 12.
12.
Okay, the luteinizing hormone levels increase. And they peak at day 14. And then there's a sharp decrease after day 14. Happy?
Yes.
That's all they wanted. They're asking you to read what you see here. So we have a fairly constant amount of luteinizing hormone until day 12, 13. I would say day 12. It's approximately day 12. Then there is a sharp increase in the luteinizing hormone up to day 14. And then there is a sharp decrease of luteinizing hormone where it evens out again. And there's a very, very ever so slight decrease by day 28. That's it. You're reading what you see.
Yes.
So that's 1 mark, that's 1 mark, that's 1 mark, that's 3 marks. So there's 6 marks here just by opening your eyes. Isn't that easy? I said to you in the beginning, if you know your graphing, you can read off a graph, you understand how to write and draw graphs. You've got 30% of your paper covered already without having opened a book. All right, now, describe the relationship between the level of estrogen and the endometrium from day 7 to day 14. So it's estrogen levels and the endometrium from day 7 to 14. So let's look. We want it from this period here, from day 7 to day 14. We're looking at this endometrium getting thicker, and we're looking at our estrogen level increasing.
Yes.
So what is the relationship here? What can you see? As the estrogen level increases from day 7 to day 14, so the lining of the endometrium thickens and increases.
Yes.
And so too, clearly, will be the glandular structure of the endometrium and the blood supply. Because remember, the thicker it gets, the more glandular it gets, the more blood supply you have. Because what are we doing? We're preparing the uterus so that should a little egg cell get fertilized, that little egg cell goes jiggy, 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 and it's got a nice, warm, spongy place to settle so that the placenta can form. You follow? The body is preparing the uterus so that it can have an egg, or at least a fertilized zygote implanted, and it can then support that little thing to grow. You with me? Okay. All you're doing there is looking at it and explaining what you see. It's another 2 marks for nothing. No learning, it's what you see. Explain why it is necessary for the level of progesterone in the blood to increase after ovulation. Now remember, what does progesterone do? What do you think progesterone does?
Progesterone, I think it's the one that makes the uterus stay thick.
It makes sure,
the uterus lining.
or let's say it maintains the thickness. The thickness of the endometrium.
Or the endometrium.
Yes, it makes sure that it stays like that. It's no good estrogen goes to all the effort of making sure that this lining builds and that it's all nice and glandular and a good blood supply. And progesterone doesn't do that. So progesterone's main function, its main function is to ensure that it maintains the thickness of that endometrium to make it ready for implantation. Estrogen got it to thicken. Progesterone makes sure it stays thick. You follow? But the other big function of progesterone is that it stops your follicle-stimulating hormone from coming about if pregnancy occurs. So if fertilization occurs and implantation occurs, it is going to be your progesterone that's going to inhibit the release of follicle-stimulating hormone. Because remember, what does follicle-stimulating hormone do? It causes...
it prepares the follicles.
It causes menstruation. It causes the lining to tear away and the formation of the new follicle. But that follicle will only start forming when the lining is torn. It's taken away. Because it means, oh dear, fertilization didn't occur. We now have to clean everything out and start again. And that's literally what menstruation is. The lining tears away. It's cleaning the surface so a new lining can develop just in case this month we are successful and we get pregnant. Because remember, your body wants you to procreate. It wants you to procreate. It wants you to produce little offspring. And it prepares every 28 days, it prepares that uterus to accept a little egg that's been fertilized. You follow?
Yes, I'm here.
Okay. It's not quite what we all want, but that is the nature's way of dealing with things. All right. So why is the progesterone in the blood increased after ovulation? Because it maintains the thickness of the endometrium, but it also inhibits the release of FHS. And remember, progesterone is only released by the corpus luteum. When fertilization does not take place, the corpus luteum disintegrates, which means, guess what? There's no more, or there's not a hell of a lot of progesterone left. So follicle-stimulating hormone isn't inhibited. It can now start going crazy again. New follicles develop, estrogen is released, and the whole process starts again. Did fertilization take place on the 28th day cycle? In the 28-day cycle, how do you know?
Because the progesterone levels decrease.
That's good. Your progesterone, and the fact that the progesterone level decreases means, sorry for you, it didn't happen. But also, if you look here, what happens to your Graafian follicle? Look here. It disintegrates. You with me? Happy, hun?
Yes, I'm here.
Okay. Explain your answer. Well, I've just done that. If you look here, your progesterone levels decrease. which means your FHS is no longer inhibited. And why does your progesterone level decrease? Because the corpus luteum disintegrates. It's the corpus luteum that releases the progesterone. Okay, happy?
Oh, very happy.
Perfect.
Thank you so much.
Okay, my darling. But it's good to look at all 4 of these graphs together. A lot of people battle with this. And all you've got to remember, follicle stimulating hormone causes the follicles, which causes estrogen. Your luteinizing hormone causes the corpus luteum, which causes progesterone. And what do they all do? They're there to make sure that uterine endometrium lining is perfect. And if it's not fertilized, well, get rid of it and start again. Okay?
Okay. Yes.
Good luck for those exams. All right.
Are
you feeling confident?
Thank you very much. Sorry?
Are you feeling confident?
I am so confident. I know I'm going to beat it. I'm so excited about it.
Good, good. You go do it, because this is lekker. And you know what? We're going to have even more fun on Sunday when we do evolution and we do environment, because that is a walk in the park. This is your hard section, a walk in the park for Sunday. Okay. Okay, people, it seems like we're on our last question. There isn't a lot of time left. We're into our last 15-minute stretch, and sure, I have to say the 3 hours have just gone just flown by. All right, we have Mpumi on the line. Are you there, hun?
Yes, I'm here.
You sound like you're 20 million miles away, Mpumi. Talk into the phone, darling. Where
Yes,
where you calling from?
I'm calling from Guyana.
From Guyana.
Yes, it's here at Limpopo.
Also Limpopo.
Yes.
All the Limpopo learners are working hard tonight. Okay,
Yes.
You wanted question 3 and it's from a prelim exam. All right,
Yes.
It says the diagram below represents a cross-section has been cut through, view of a human seminiferous tubule. In other words, it's in the male because we only find seminiferous tubules in the male, in the testes where the sperm are produced, okay? In which spermatogenesis is occurring. Now, people, remember that gametogenesis is the formation of the gametes through the process of meiosis. All right? It's the different stages that it undergoes in order to produce haploid gametes. So in the male, it's called spermatogenesis, and we produce sperm cells. And in the female, it's called oogenesis, and we are going to provide or produce the egg cell. Okay, here we go.
Okay.
So this is what this diagram is. So it shows you the connective tissue between the adjacent seminiferous tubules, because remember the seminiferous tubules are all interwoven. It's like if you took a ball of wool and you unraveled that ball of wool and you just made it into a clump this like that, that is what those seminiferous tubules look like, okay? So it's just like a big mush of wool. Now each wool piece would be the seminiferous tubule. Now in there we're going to find the cells of Leydig, and they are important. Why? Because they produce testosterone. Okay, testosterone. Okay. That's the male hormone. That's what makes guys all their secondary characteristics. And if women take testosterone, they end up with big muscles as well. All right, they're not supposed to have it. But that's your testosterone that is produced by the cells of Leydig. Okay, then we get the spermatogonium, and the little spermatogonium will undergo meiosis 1 and meiosis 2, and they eventually end up forming the spermatids, and the spermatids will mature, and they form the sperm cells. Don't just call it sperm, it's sperm cells, all right?
All right.
You happy with that? Okay.
Yes, I'm very.
All right, now, here we go. Name the hormone produced by the cells of Leydig. Well, I've already told you it's testosterone.
Yes.
Okay? Name one function of this hormone. It's for the secondary male characteristics. Okay, it's for all secondary male characteristics. Facial hair and the big muscles and hair in the pubic area and under the arms and etc., etc. That is the voice dropping. Those are your secondary male characteristics. How many chromosomes are in each of the following? Now, if you look at the spermatogonian cell and the spermatid, in your spermatogonian cell, now remember how many chromosomes. Remember, in humans, we have 23 pairs of chromosomes, or we can say we have 46 chromosomes. Okay, that's in males and females. That's what we have in our cells. That is if it is diploid. Okay? So, in your spermatogonium, it hasn't undergone meiosis yet. So, therefore, it is going to have 46 chromosomes. Or you can say 23 pairs. Or you can say a diploid chromosome number. In your spermatid, meiosis has occurred. So they are going to have 23 chromosomes. You with me?
Okay, so, yes, I'm listening.
So this is normal. That's what you have. It's what I have. In our nuclei of our cells, we have 46 chromosomes or 23 pairs of chromosomes in every single nucleus in our body. Okay?
Okay, so does it mean that on spermatogonium is when all our chromosomes are added, like?
No, no,
That's
no.
what I'm saying.
No, 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 no.
One there-
Listen, in every cell in your body, you have a nucleus. Okay, look here.
Yeah.
There's the cell. Or in fact, let's make space. Here's a cell. Okay, in that cell, you have a nucleus. In the nucleus, you have chromosomes.
Okay.
Okay? How many
Yeah.
chromosomes? You are going to have 23 pairs of chromosomes. Now, if I take 23 pairs, like say you got 23 pairs of socks and I split them open, I'm going to have 46 chromosomes.
Okay.
And those 46 chromosomes pair up to 23 pairs. You follow?
Yes. I follow.
Okay. Now, here, that's normal. Your spermatogonium is normal. It's a normal cell. It hasn't undergone meiosis II yet. And it hasn't undergone, meiosis is about to happen there. So meiosis occurs, your spermatid, which grows into your sperm cells, it's already undergone. So here, meiosis. And once meiosis has occurred, your spermatid then ends up with 23 chromosomes. It's half. It's N. It's
Yeah,
haploid.
That's why I had a problem.
Okay. You happy with this now?
I'm very happy.
Okay. Then explain the importance of the spermatogenesis as a mechanism to introduce variation by referring to prophase 1 and metaphase 1. Now, you've got to remember that during prophase 1, you have crossing over. We spoke about the crossing over earlier.
Yes, I was watching that.
Okay, where you have your chromosomes, and the chromosomes come next to each other like this. Okay, and they lose pieces. So if this one is all shaded, okay, and this one is all white, they then end up looking like this. This little piece will be there, and this one will be white, and everything else will be shaded. You follow?
Yes,
And
I
the
follow.
fact that they are sharing genetic material, that we say it is, that helps for genetic variation. You with me? So
Yes, I'm here.
that's your genetic is in prophase 1. Why? Because of the chiasma, and the chiasma is where they touch, and the process is called crossing over.
Yeah.
You with me?
Yes, I'm here.
Okay, my darling. Now, metaphase 1, okay, I need space. Metaphase 1, remember you're going to have your chromosomes lined up like this in metaphase 1. All right? There's the equator. And maybe this one and this one go this way. And this one and this one go this way. Or maybe this one goes up and this one goes down. You follow? So this little piece goes this way and this one goes this way. So what you have here is independent, and I'm not showing this properly, independent assortment. But it means that when I've got these chromosomes lying here on the equator like that, okay, I can end up with this one going this way, and that one going this way, and this one may go down, and this one may go up. Each chromosome will separate independently.
Okay.
And that is why metaphase 1 is so important, because each chromosome, as it separates out, and it now halves, so it goes from being your pair, going from diploid to haploid. This chromosome, the one half will go up and the other half will go down, or the one of the pair goes up and the one goes down. Here, this one may go down and that one may go up, but they split differently. So each chromosome separates independently. You follow?
Okay.
You've got to remember that of your chromosomes, for every characteristic, you're going to have, if you look at this here, you've got 2 chromosomes. One came from your mother and one came from your father. So we must have them crossing over so they share, and we have genetic variation because they share genes. And we must make sure that for every chromosome set, not all your dad's chromosomes that you got from your dad goes one way and all your mom's go one way. In some cases, your dad's one will go up to make the one sperm cell, and your mom's will go down to make another sperm cell. So that's why metaphase is important. And listen, my darling, I've got to say goodbye. That's it. We're sort of done here. All right.
Oh, thank you so much.
Okay, my hun, I hope you understand better now.
No, I do.
Good. And to all the learners out there, our Learn Extra team here wishes you the best of luck. Please make sure that you go to bed. I'm not saying go to bed at 8 o'clock, but go to bed early tonight. Make sure you sleep well. And this is for every exam. Make sure you get a good night's rest. Don't get up early and try and cram everything into a couple of hours. Perhaps tomorrow morning, get up at 5, 5:30, read through your summaries. That's all. Just to settle your nerves a bit. Have a good protein breakfast. Now, please remember that on Sunday night or Sunday afternoon from 4:30 to 7:30, we're doing Life Sciences Paper 2. We'll be going through evolution, and then accounting is going to be on Monday. I hope I've got that right. But it's accounting on Monday evening, so make sure that you tune in, and we'll take you through all of that. We'll give you the confidence you need to go in there and just slay that dragon. People, please have a good breakfast. You can't write exams when you're half sleepy because you have got no blood sugar. You learned this last year. You've got to keep your blood sugar constant, and please read your questions carefully. And tonight, if you do anything, make sure you are able to graph, that you can draw a line graph if you're given the information, that you can put together a table of a record of results from a bar graph, that you are able to draw a pie chart. Make sure that you can do those silly things because they will give you 30 to 40% in an exam. They're silly, silly things. Good luck, goodbye, and enjoy. Bye-bye.